So here we see a slide showing the cognitive trajectories of the two arms in COCO, the coached arm in the blue dotted line and the standard of care arm in the solid blue line. And I fit them with a spline curve. So one of the things to notice is the response is nonlinear. This is pretty typical for Alzheimer's interventions all the way back. If you look at the original results for like drugs like donepezil, you see an initial improvement. In some cases, in some trials, you'll see that improvement fade away over time. In this case, you see it's sustained improvement, but it doesn't continue to grow like it does in the early months of the trial. But it's sort of remarkable. In some sense, we see for the intervention arm, it deviates very little from baseline. We have a relatively small number of people in the trial, so we're not 100% confident exactly where the standard of care and the coaching trajectories are with respect to, say, baseline, but we know they're significantly different. The significance here is very significant for the difference in the two arms. And at 18 months, which is about here, you see about a 60% reduction in cognitive decline. So this is an impressive result. We see this. I've shown you the trajectories in a nonlinear form because I think that's what the data are actually showing us. Our actual statistical testing is done with linear regression because that's the standard formal way of doing statistics. So anyway, we think this is adds to the existing body of knowledge that multimodal interventions are effective treatments for Alzheimer's disease. Let me start by just giving you a rough sense of what systems biology is trying to do from a knowledge generation or an epistemology point of view. So most clinical trials measure relatively few data points. Some only measure a couple, like cognitive outcome. And that would be like looking at a digital camera image with just 1 or 2 pixels. So they might look at sort of this top row and say, oh, look, we're giving people a drug here at baseline. And by the end of the trial, there's a few more pink dots, and we can do a statistical test and say there's more pink dots, and therefore our drug is working. As you gather more data, which we do, we measure the thousands of proteins and metabolites, as well as a number of neurocognitive outcomes. As you gather more data, in this case, a picture of a rose, at some point, even though there's just sort of an incremental quantitative increase in data, at some point the human brain looks at these data in a completely different way. It's a qualitative change. So by the time you have enough data to see the whole picture of what's going on, you're not asking questions about the number of pink dots. You're saying, wow, this is a pretty rose. When should I water it? And those are just completely different types of questions. And that's what we want to do with Alzheimer's research. We want to get enough data and enough mechanistic understanding so that when we see all the data together, we see a picture, and it brings to us concepts like this is a rose. And I'm actually not quite there. I'm somewhere in the middle, but I think I'm getting there to sort of see pictures. So if I look at all of the molecules in the blood, so in this case, I'm looking at 1,400 molecules, and display them in a way that this is a dimensionality reduction display, I can point out those molecules, those analytes that change in response to intervention. So just by calling somebody up on the phone, I can make all of these molecules that are big dots on this slide in your blood change. Now, obviously, I have expert coaching. I'm using coaching designed to get behavioral change. And this, presumably, it's not just receiving the phone call. People are actually doing the exercise and dietary changes. And they're not random. They're clustered in pathways. And there's a large number of them, which you kind of expect. This is big. If you start changing your diet and exercising a lot, you might expect a lot of molecules in very specific pathways to change. And so this starts to give me a picture. And there's a number of ways as a systems biologist, I can sort of put these points together to see a picture. And one of them is a correlation network. And that's what this is. And when I do this and look at not just those molecules that change in response to the intervention. So obviously, if I tell you to diet and exercise, a lot of things are going to change in your blood, but not all of those things are going to cause your cognition to ameliorate or prevent dementia. So I also want to know which of those molecules are correlated with cognitive improvement, beneficial, or in some cases, not so beneficial outcomes, which are correlated with change. And then I can look at how they relate to each other in an operational network like this one. And they tend to cluster. And so now I get to see pictures like, is this part of a rose here, or is this part of a rose down here? This is actually an immune response subnetwork, and implies that there's some aspect of the immune system, inflammation or immunity, that's mediating the intervention and presumably ameliorating cognition. And these are probably related to diet, but also, curiously enough, are connected to the immune system. So it seems like what we're seeing here, if we look at the molecules in this sort of more diffuse subnetwork, evidence of reduction in caloric intake, which makes sense for a typical American population, and probably reduction in red meat consumption as well. So not only, this comes back to my original approach to trying to generate knowledge, is I don't just want to say, yeah, a multimodal intervention works, because as soon as I say that, people are going to say, well, exactly what diet? How does it work? Is it all parts of the multimodal intervention, or just the exercise, or just the diet? So I really need to get into a mechanistic understanding to tell you what to do next, and tell researchers what to do next. What should we test next? Because presumably what I did is not the very best intervention. There's got to be ways to improve it. We think that one of the things that getting dense data on trials allows us to do is do a little bit quicker trials. So by seeing these biomarkers, we can understand what's happening in less than, say, 10 years of following people over the full cognitive decline. And then that allows us to tweak both or understand how our interventions worked and how we might tweak them to do new trials quicker. So we have another trial, which we started after COCO, and it's now about halfway through, called the prevention trial. And it's not fully informed by COCO because we started it before COCO was done, but we wanted to test sort of a variation. So COCO is sort of a light touch. I call you up and say, hey, exercise more, change your diet. There's a little more than that. But prevention is an in-person, where you have in-person diet coaching, in-person exercise coaching. So it's kind of intense. And so but roughly speaking, we're testing the same thing with dense dynamic data. This is all nonprofit funded. So one of the things we would like to do is see bigger trials that are more robust and can get more information. Our trial was also substantially impacted by COVID. So we ended up enrolling fewer people than we wanted. And so we'd like to do more of the same, both on Alzheimer's disease and on other dementias and other neurologic diseases. And for that matter, all sorts of clinical trials across all of medicine.